Genomic selection has developed since the mid-1990s from a hypothetical concept to a practical way of applying genetic marker data in breeding. As the name suggests, the objective of genomic selection is to provide breeders with a method of making selections in breeding programs. This stands in contrast to statistical approaches, such as association genetics, that were initially developed to identify specific genes or even specific DNA sequence variants as the causative agents underlying phenotypic variation. Association genetics approaches have also been applied in some breeding programs, but their primary use has been in human biomedical genetics to study the genetic basis of diseases. This module provides some historical perspective of genomic selection and an overview of different approaches to the computational problems of estimating breeding value from marker genotypes, followed by an example of successful application of genomic selection in dairy cattle breeding, and a discussion of strategies that may be useful in applying it to forest tree breeding. One early and visionary publication that outlined the possibility of making selections based on marker genotypes from very dense genetic markers across the entire genome was that of Haley and Vischer. Other earlier publications had outlined some of the same concepts, and Haley and Vischer noted in particular the contributions of Charlie Smith in a 1967 paper and in a 1993 abstract. The key insight that pushed these pioneers to consider new ways of using marker data was the realization that the number of genetic markers would not always be limiting. It is important to note that the technological capability to discover and genotype high-density markers did not exist for crop, livestock, or forest tree species in 1998 when this paper was published. But the authors had the foresight to see that high-density marker genotyping technology could be developed and would have a major impact on the utility of markers for breeding programs. Another insight, also based on the assumption that marker genotype data would no longer be limiting, was that marker information would supplement or perhaps completely replace the use of recorded pedigrees, because the relationships uncovered by high-density marker information would provide more detail about kinship than the estimates calculated from known pedigree relationships. Another insight of the authors was that high-density marker genotypes could account for the many loci with effects too small to reach statistical significance, which nevertheless do contribute to the genetic control of traits of commercial interest. This is a point of divergence from methods developed primarily to identify genes that underlie phenotypic variation, because it means that testing the statistical significance of an association between a marker and a trait is not a concern for genomic selection. Instead, genomic selection attempts to build a statistical model that has power to predict phenotypes from genotypes, then uses that model to make selections among individuals for which genotypes, but no phenotypes, are available. Haley and Vischer also noted out that accurate and precise phenotype data would continue to be central to breeding operations because those data are essential in creating models that divide phenotypic variation into its genetic and environmental components. Methods used for analysis of QTL experiments, in which the number of genetic markers is typically lower than the number of individuals for which phenotypic and genotypic data are available, are not suited to analysis of the very high density genetic marker data required for genomic selection. Similarly, statistical models used in association genetics are not well suited to genomic selection either, because those models typically test each marker locus separately for an association with phenotype and require that any population structure or relationships be explicitly considered in the model to avoid false positive associations. Mewison, Hayes, and Goddard proposed and tested two different approaches to this problem, using simulated data because high-throughput genotyping methods had not yet been developed for agricultural and forest species. The first approach assumes that all markers contribute equally to the genetic variation in phenotype, so the problem is reduced to estimating a single variance rather than estimating as many variances as there are markers. An alternative approach is to assume that each marker locus can explain a different fraction of the total genetic variation, and that most of those fractions will be zero or close to zero, while only a small subset of markers will explain a fraction of the genetic variation that differs from zero. This approach requires the use of Bayesian methods to estimate the individual marker variances and is computationally demanding. The basic procedure is to assemble a training population of individuals for which both genotypes and phenotypes are available, and use those data to create a statistical model that relates variation in observed genotypes of marker loci to variation in the observed phenotypes of the individuals. Multiple generations of parents and offspring provide a more powerful training population than a single generation of individuals, and larger numbers of individuals, generations, and markers provide more powerful training populations. The statistical model is then applied to a prediction population comprised of individuals for which genotypes are available, but phenotypes are not. Genomic selection is based on similarity between the training and prediction population in the linkage disequilibrium between marker loci and trait loci. This similarity may exist because the prediction population is descended from the training population, or because the density of markers is so high that every trait locus is in disequilibrium with at least one marker locus across the entire population of the target species. Cross-validation is a method of testing the predictive accuracy of the statistical model developed using the training population before trying to apply the model to predict breeding values of the individuals in the prediction population. The training population is composed of individuals for whom both phenotypes and genotypes are available, so dividing it into subsets creates two populations with known genotypes and phenotypes. The statistical model can be trained on one population, then used to predict the expected phenotypes of individuals in the other population. The predicted phenotypes are then compared with the observed phenotypes to assess the predictive accuracy of the statistical model. Ideally, the relationship between the two subsets of the training population should be similar to the relationship between the training and the prediction population. If the relationship between the two subsets of the training population, that is, full siblings, is different from that between the training and prediction populations, that is, parent-offspring, then the predictive accuracy observed in cross-validation studies may not accurately reflect the accuracy that will be obtained in predicting the phenotypes of the prediction population. As an example of training and prediction populations, consider the following scenario. 100 grandparent individuals, labeled GP1 through GP100, are crossed in pairs, and 50 offspring are produced from each of the 50 crosses. One individual is selected from each of those 50 families for use as a parent, labeled P1 through P50 here, and the full sibling individuals within each family, labeled FS1 through FS2500, are also used for collection of SNP genotypes and phenotypic data. The 50 parents are then crossed in pairwise combinations to produce 25 families of 100 progeny each, and phenotypes and genotypes are collected for all 2,500 of those individuals as well. Together, these 5,100 individuals, grandparents, parents, full SIB parents, and offspring, constitute the training population. Suppose now that 50 selections are made among the 2,500 offspring, and the selected individuals are crossed in pairwise combination to produce 100 progeny per cross, labeled Gen 2-offspring. These individuals are still so young that no phenotypes have been measured, but DNA samples have been taken and SNP genotypes determined. These individuals constitute the prediction population for the purposes of the breeding program. These are the individuals for which breeding values are to be predicted by the statistical model. Cross-validation is a method of evaluating the predictive accuracy of statistical models developed by different methods, so that the method that yields the most accurate model can be used with the entire training population before trying to apply the model to predict breeding values of the individuals in the prediction population. One way to evaluate the accuracy of different models is to use those models to predict breeding values for individuals with known phenotypes. Then compare the correlations of observed phenotypes and breeding values predicted by different models. The training population is composed of individuals for whom both phenotypes and genotypes are available. So dividing it into subsets creates two populations with known genotypes and phenotypes. The statistical model can be trained on one population, then used to predict the expected phenotypes of individuals in the other population. The predicted phenotypes are then compared with the observed phenotypes to assess the predictive value of the statistical model. This process can be repeated multiple times using various strategies of sampling the training population with or without replacement to create the training and validation subsets. Ideally, the relationship between the two subsets of the training population should be similar to the relationship between the training and the prediction population. If the relationship between the two subsets of the training population is different from that between the training and prediction populations, then the predictive accuracy observed in cross-validation studies may not accurately reflect the accuracy that will be obtained in predicting phenotypes of the prediction population. In this case, the training population for cross-validation includes the grandparental and parental generations, and the validation population consists of the progeny generation. The same relationship will exist between training and prediction populations if the parents and the offspring are used for training a model to be used in predicting the breeding values of the Gen 2 offspring individuals. The training population still consists of grandparents, parents, and aunts and uncles, shown in purple, of the individuals in the prediction population, shown in red. The 100 individuals in the grandparental generation, GP1 to GP100, are shown in gray, because in principle they need not be used in this scenario. In practice, using those 100 additional breeding values would probably increase the accuracy of the final model, and there is little reason to exclude them. One simplifying assumption that can be made is based on the traditional quantitative genetic model that continuously varying traits are under infinitesimal genetic control, that is, controlled by many loci, each with equally small effects. This assumption assigns each marker an equal proportion of the total genetic variance, so that instead of estimating as many variances as there are markers, a single value for the total genetic variance is estimated and then divided equally among all markers. This approach to genomic selection is functionally equivalent to using marker genotype data to estimate the realized genomic relationship matrix, followed by best linear unbiased prediction of breeding values based on phenotypes of relatives. The data from quantitative trait locus mapping experiments in livestock and annual crop plants suggests that the assumptions of the infinitesimal model are not always correct. QTL mapping experiments sometimes detect loci with relatively large effects, accounting for more than 10% of the genetic variation in a particular trait, and at the same time also detect loci with smaller effects. This might be termed an oligogenic model, in which several genes are involved in genetic control of the trait, as opposed to the polygenic model that assumes many genes are involved. An alternative approach to genomic selection, consistent with the oligogenic model of inheritance, is to allow each marker locus to have an independent variance, but to assume that most of those variances will be 0 or close to 0, while only a small subset of markers have variances that differ greatly from 0. Best linear unbiased prediction, or BLUP, is a statistical method originally developed for animal breeding. The method uses information about the phenotypes of relatives, as well as the observation of an individual's phenotype, if that is available, to estimate the genetic value of that individual. For some traits, much of the phenotypic variation is due to environmental effects rather than genetic variation, so the average phenotype of related individuals can be more accurate estimators of the average genetic value of individuals than the individual phenotypes themselves. This type of trait would have low heritability and a small genetic variance relative to the residual variance. The shrinkage factor would be large, and the phenotype of the individual would be less important than the mean phenotype of the population in estimating the true genetic value of the individual. The phenotype of the individual may be more extreme than its estimated genetic value, and the shrinkage factor is so named because it reduces the difference between the estimated genetic value and the mean of the population relative to the difference observed between the individual's phenotype and the mean of the population. This method works well, particularly when the related individuals used to estimate the genetic value of an individual are progeny of that individual. Bayesian methods often use a Markov chain Monte Carlo process to estimate parameters for which exact values cannot be computed. A Markov chain is a series of states, or sets of values, of experimental parameters, in which the state at a given step is dependent upon the state at one or more previous steps. Monte Carlo refers to sampling parameters at random from a distribution, its name for a famous gambling resort in Europe. So, the Markov chain Monte Carlo methods explore the parameter space of possible parameter values to determine which values are most likely given the observed data. Exploring the sample parameter space requires methods that move from one state to another in an efficient way. The Gibbs sampler and the Metropolis-Hastings algorithm are two among many ways to do this. The ideal outcome of a Bayesian analysis is to produce a set of posterior distributions of parameter values, one for each of the parameters to be estimated, in which there are clear modal values demonstrating that one value of the parameters is most likely given the observed experimental data. Another aspect of the ideal outcome is that the modal values of the posterior distributions should be independent of the prior values chosen as the starting points for the Markov chain. Mewison and co-authors tested 3 approaches for analysis of genetic variance using genotypes at thousands of marker loci and compared those three approaches to a standard least squares method. One of the three approaches was based on BLUP and required the simplifying assumption that each marker or interval between markers explains the same fraction of total genetic variance. It is important to note that markers are treated here as random effects, so the number of markers used to explain phenotypic observations can be much greater than the number of phenotypic observations to be explained. One consequence of this is that the effect of each genetic marker is estimated as a genetic variance, rather than as a known or fixed effect on phenotype. Two Bayesian approaches with slightly different assumptions were tested. Bayes A used a prior distribution with an infinitesimally small probability of zero variance, meaning that while markers or intervals could have different variances, these variances were unlikely to be exactly zero. Bayes B added the possibility that the variance of a given marker could be zero, but the proportion of markers with zero variance was assumed to be known and had to be provided as a parameter value before conducting the analysis. The assumptions underlying different statistical models also have implications for the number of markers required to carry out genomic selection in a breeding population. Predicting genetic value based on the infinitesimal model is very similar to standard BLUP methods for estimation of breeding values based on phenotypes, and requires only that the marker data be informative enough to estimate the proportion of alleles shared among related individuals. The exact number required is dependent on the number of chromosomes in the target species, the size and diversity of the breeding population, and the degree of relationships within and between the training and prediction populations. Fewer markers are required for target species with fewer chromosomes, as well as for target populations with less genetic diversity and higher relatedness among individuals in the training and prediction populations. In effect, the marker data in this situation are reconstructing the relationships as shown by the pedigree, with the additional advantage that marker genotypes can resolve Mendelian segregation within full sibling families, which traditional pedigree-based BLUP methods cannot. Predicting genetic value based on an oligogenic model builds on the information provided by reconstruction of the pedigree and uses additional information from linkage disequilibrium between specific marker loci and specific trait loci. The amount of the additional information available depends on the true pattern of genetic control, as well as the extent of linkage disequilibrium between markers and trait loci. Most forest tree species are not highly inbred and have relatively large effective population sizes with low levels of LD. These are characteristics that increase the number of markers required to assure that every trait locus is in LD with at least one marker. In contrast, populations of annual crop plants such as barley can have high levels of LD due to inbreeding and limited effective population size. A few thousand marker loci may be sufficient to cover the genome adequately for purposes of genomic selection using Bayesian models. The best statistical method to use depends in part on the underlying genetic architecture of the trait. Breeding values for a trait that is controlled by a few major genes will be more accurately predicted by Bayesian methods, while breeding values for a trait that is controlled by several hundred genes of equally small effects should be predicted equally accurately by either Bayesian or BLUP methods. BLUP methods are often significantly easier and faster in computational terms, so there is some incentive to use BLUP methods if they will work well. Some traits can be expected to be controlled by smaller numbers of genes, with the potential to have individual genes with major effects on phenotype. For example, the DGAT1 locus on bovine chromosome 14 was shown by QTL mapping experiments to explain a substantial amount of variation in the fat content of milk in dairy cattle, and this locus has also been repeatedly detected in genomic selection experiments. More complex traits, such as growth in forest trees, are likely to be affected by variation in hundreds or even thousands of different genes, and so the infinitesimal model may describe the underlying genetic architecture fairly well, thus allowing use of BLUP methods. Domesticated plant and animal species used in agriculture have often undergone a domestication bottleneck, meaning that much of the genetic variation detected in wild relative species is absent from the domesticated species. This reduction in genetic diversity presumably occurred over the thousands of years of selected breeding prior to development of the modern understanding of genetics, and was caused by repeated selection for desired phenotypes over many generations, coupled in some cases with mating of relatives. Few, if any, forest tree species have undergone similar selective breeding for an equivalent number of generations. For the most part, tree breeding programs are still working with populations that are quite similar to those found in natural stands. While the effective population sizes vary for different species of forest trees, in general they are likely to be larger than those found in domesticated annual crop and livestock species, and linkage disequilibrium is likely to decay over much shorter distances in tree genomes than in genomes of domesticated agricultural species. The greater genetic diversity and lower levels of LD in typical forest tree populations mean that much higher densities of marker loci will be required to obtain markers in LD with every trait locus across the entire breeding population. Genotyping resources also differ among forest tree species. Reference genome sequences are available for poplar and eucalyptus, so high-density SNP arrays can be synthesized based on known physical locations of specific loci. As of 2011, few conifers have a comparable level of genomic resources. Genome sequencing projects are underway for several conifer species, however, and high-density SNP arrays will probably be available by 2015 or before. Linkage disequilibrium, or the utility of the allelic state at one locus to predict the allelic state at a different locus, is strongly dependent on allele frequencies at the two loci under consideration. The next few slides will explore this dependence in mathematical terms. The objective is to demonstrate the challenge facing breeders who would like to have marker loci and strong LD with every locus that has a genetic effect on every target trait in the breeding population. Achieving this objective essentially requires that several marker loci with different minor allele frequencies be present in every region of the genome, so that no matter what the allele frequency may be at any nearby trait locus, at least one marker locus will have a minor allele frequency that is similar to that of the trait locus, and is in LD with it. Linkage disequilibrium can arise as a consequence of the evolutionary history of mutations in a population, or as a result of selective pressures. Drift, non-random mating, are other factors that influence allele frequencies. For the sake of simplicity, the following example will consider only the evolutionary history of two loci, A and B, and two mutations to alternative alleles, one at each locus, giving rise to lowercase a and lowercase b alleles. In scenario 1, the mutation of uppercase B to lowercase b occurs on a chromosome that bears the uppercase A allele, so that every copy of the lowercase b allele descended from that original chromosome is always paired with an uppercase A allele, never with an lowercase A allele. In scenario 2, the mutation of uppercase B to lowercase b occurs on a chromosome that bears the lowercase A allele, so that every copy of the lowercase b allele is paired with a lowercase a allele and never with an uppercase A allele. In both scenarios, the mutant alleles, A and B lowercase, are assumed to be the minor alleles, which is to say they have frequencies of less than 0.5 in the population. In the first scenario, the minor allele, lowercase b, occurs paired with the major allele, uppercase A, and linkage disequilibrium is quite low across most of the possible combinations of minor allele frequencies. Alleles lowercase a and b are defined as minor alleles, and so the maximum value of possible frequencies for those alleles is modeled as 0.49. The three-dimensional plot on the left side of the slide shows linkage disequilibrium as r squared, calculated using the formula on the previous slide, for combinations of minor allele frequencies ranging from 0.01 to 0.49 for lowercase alleles A and B. This plot shows that LD between alleles uppercase A and lowercase b is highest in the case where the allele frequencies of all four alleles approach 0.5, producing a peak at the back corner. The histogram on the right shows the distribution of LD, again expressed as r squared, across the range of possible values between 0 and 1, calculated from 2,304 possible combinations of allele frequencies ranging from 0.01 to 0.49 for the minor alleles A and B. The frequency of LD values between 0 and 0.2 is much higher than the frequency of LD values greater than 0.2. We can conclude that minor alleles at SNP marker loci have the most predictive power regarding the presence of a major allele at neighboring trait loci when the allele frequencies at both loci approach 0.5. As the allele frequency at either the trait locus or the SNP marker locus diverges from 0.5, the predictive power of the SNP genotype drops and is relatively low for most combinations of allele frequencies at the marker and trait loci. In scenario 2, the minor alleles are linked in coupling, or paired with each other on the same chromosome homologue. In this case, the frequencies of the minor alleles can be equal across a range of allele frequencies from 0.01 to 0.49. The three-dimensional plot on the left shows that LD, expressed as r squared and calculated according to the equation presented on slide 19, is equal to 1 along the diagonal line where the frequencies of the lowercase a and b alleles are equal, but as the frequency of the lowercase b allele drops below that of the lowercase a allele, LD drops very quickly. This effect is most pronounced if the frequency of the lowercase b allele is low and the frequency of the lowercase a allele is moderate to high. The strong dependence of LD on allele frequencies means that LD will often be low between rare mutations at trait loci that cause changes in phenotype and SNP marker loci with high minor allele frequencies that are commonly available in commercial genotyping arrays. The distribution of LD values shown in the histogram on the right is much different than that shown on slide 20, because many more combinations of allele frequencies can give rise to LD near 1, but there are still many combinations of minor allele frequencies that yield LD levels less than 0.2. The example scenarios showed how LD changes with differences in allele frequencies among SNP marker loci, typically chosen to have relatively high minor allele frequencies, and mutations that cause differences in phenotype, which may be rare alleles with very low minor allele frequencies. In a breeding program, however, allele frequencies can be manipulated by structuring the mating design and size of a reading subline. For example, consider a subline of 25 individuals mated to each other in a partial diallel design with 4 families per parent and 10 progeny per family. The result will be a set of 1,000 progeny, but the lowest minor allele frequency in the population should be around 2%. The 25 original parents have a total of 50 alleles, and the minimum frequency would be a rare allele present as a heterozygote in only one of the 25 parents. This rare allele should still be present in half the progeny of that parent, or about 20 offspring, because each parent contributes to 4 families of 10 progeny each. Any individual selected at random from a wild or nearly wild population of forest trees may be heterozygous for thousands of alleles that are extremely rare in the population as a whole, but within progeny of a 25-parent subline, the lowest expected value of a minor allele frequency is 2% for all of those thousands of rare alleles. A subline initiated with 10 founding parents would have a minimum expected minor allele frequency of 5%. Linkage disequilibrium between SNP marker loci available on commercial genotyping platforms and a trait locus can be very different in populations of progeny from breeding populations with very small effective population size than it is in the entire population of the species. N sub E is the term for effective population size, or the hypothetical number of unrelated individuals that would have the same genetic diversity as the observed population. In a tree breeding program, a subline might be made up of 20 individuals crossed to each other in a structured mating design. Such a mating design might give rise to a population of progeny with a census number of 1,000, but the effective population size is no more than 20, because the only alleles present in the 1,000 offspring are those present in the original 20 parents, neglecting the minor effect of new mutations that occur during this one generation interval, of course. Identity by descent, or IBD analysis, is a term associated with linkage analysis within human pedigrees, but in tree breeding programs, pedigrees can be much larger and more interconnected than in most human families. The distinction between linkage disequilibrium methods in small populations and IBD analysis in large pedigrees disappears as the size and genetic diversity of small populations approach those of large and complex pedigrees. The essence of genomic selection is modeling the effects of genotypes of non-observed loci, that is, those that actually cause phenotypic variation, based on the genotypes of observed marker loci. So the report by Powell and co-authors of mathematical approaches specifically targeted at this objective is quite relevant to tree breeding, even though the focus of the paper is on inheritance of complex traits in human populations. This example shows 16 parents crossed in 4 pairs to give 8 first-generation offspring, which are then crossed to yield a total of 16 second-generation offspring. Each homologue of the single chromosome shown for each parent is a different color, representing a different haplotype, with a total of 32 different haplotypes depicted in this image. The lowest possible minor allele frequency in this pedigree would be 1 in 32, or 0.03125, for an allele that occurs in only one of the 32 parental haplotypes, shown in different colors here. One can imagine a more complex mating design. For example, the 16 parents could be crossed in two 8-parent partial diallels as different sublines, and then first-generation progeny intermated with each other within subline to produce second-generation progeny. Regardless of the complexity of the mating design, if sufficient marker information is available to detect the ancestral haplotypes in the progeny, then the identities of the chromosomal segments inherited from parents and grandparents can be determined unambiguously. The number of SNP markers per chromosome required to detect the ancestral haplotypes increases with the number of generations, and also with the number of different parents that contribute to each line of descent. Inbreeding results in accumulation of fewer different haplotypes per chromosome homologue than outbreeding, for an equal number of generations. So breeders working with inbred species can achieve adequate resolutions of haplotypes using fewer markers than are required by breeders of outbred species. Outbred populations, in which self-fertilization occurs rarely or never, are typically quite heterogeneous in genetic terms. This heterogeneity is shaped by demographic history, so factors such as the rate of population growth, the number of individuals among whom mate selection occurs, and the extent of migration or other forms of gene flow can all have dramatic impacts on the genetic architecture of the population of interest. For example, rapid expansion of a population from a small number of progenitors to a large population size leads to larger blocks of ancestral haplotypes in the final population than does maintenance of a large population size for the same number of generations, as shown by Chapman and Thompson in 2002. For this reason, haplotype structure in human populations, which have increased in size dramatically over the past 10,000 years, often includes linkage disequilibrium to a much greater extent than is found in conifer populations that have been stable or decreasing in size over the last 10,000 years. In the same way, the evolutionary history of trait loci and marker loci influences the statistical relationship between markers and trait loci. New genetic variants can arise at any time through new mutations, and new combination of alleles created by recombination may have novel effects on phenotype as well. Statistical models based on either LD or identity by descent must be retrained every few generations to take these new sources of variation into account. One very successful example of application of genomic selection methods to applied breeding is found in dairy cattle, particularly Holstein cattle. As previously noted, the effective population of Holsteins is less than 100 worldwide. So as a result, linkage disequilibrium is quite high relative to the levels found in forestry populations. The U.S. Department of Agriculture maintains an active research program in dairy breeding, as do many other countries, and several publications have reported on the success of genomic selection applications. This slide outlines the findings reported by Van Radden and co-authors at the USDA, though other groups have reported similar success in other countries. Several elements are required for successful application of genomic selection to tree breeding or population genetic analysis, as shown on this slide. The exact numbers of individuals in the training population may vary among tree species, depending on the genetic architecture of the traits of interest and the distribution of genetic variation within the breeding population. But 1,000 individuals is a reasonable size based on simulation studies reported by Grattapaglia and Resende. These elements are available for some forest tree species, and research and development efforts are underway to explore the power of genomic selection for tree reading using the available tools. Our tool is expected to be available very soon. The yield of DNA sequence data, expressed in megabases of raw sequence data per dollar spent on reagents and instrument time, has been dropping exponentially since the introduction of automated sequencing technology in 1986. But the rate of decrease accelerated dramatically in 2005 with the introduction of new highly parallel sequencing platforms. At the current rate of expansion of DNA sequencing technology, the cost of obtaining nearly complete genome sequence data on founders of breeding populations is likely to drop to an acceptable level within the next few years for some high-value species. Mewison and Goddard used simulated data to explore the added value of having whole genome sequence information of population founders and imputing that high-density data to progeny based on identity by descent estimated from a relatively low-density set of SNP genotypes of the progeny. Those simulations showed that imputation of SNPs from founder whole genome sequences to progeny provided a substantial gain in accuracy over the accuracy obtained using currently available high-density SNP genotyping platforms. Bayesian methods may be better suited to dealing with the extremely high densities of SNP loci available from imputation of whole genome sequence information, although again that is likely to depend on the true underlying genetic architecture of the target traits. One key advantage of using whole genome information is that it is likely to capture the causal variants that actually underlie phenotypic variation, which should mean that the statistical models built from those genotypic data will remain very accurate for several generations. To summarize, the central objective of genomic selection is to predict breeding value rather than to identify specific genes. The accuracy of prediction is a function of 4 characteristics, 2 of which are under the breeder's control, and the other 2 of which are not. Breeders can control the effective population size and marker density used in their work, but have much less control over trait heritability and genetic architecture. Careful collection of phenotypic data and well-designed and controlled tests can have a positive effect on increasing trait heritability. Well-designed tests to collect accurate and precise phenotypic information continue to be a valuable asset in breeding programs. Changes in DNA sequencing technology will have profound impacts on the cost of genotyping, and experiments that are not possible today are likely to be not only possible, but very cost-effective within a few years. Eventually, breeding programs will be able to exploit complete genome sequence information, which should lead to very powerful and accurate statistical models that will be useful for multiple generations. This list includes only a brief introduction to the literature on genomic selection. Many excellent papers could not be included due to space limitations, and new papers are published every month describing further advances in genomic selection methods.